Good day, everyone. You're welcome to my YouTube channel, Everything Medical. On today's video, I'll be treating the vitamin A biochemistry, known as the retinol. Before we dive into this class properly, let's give a little bit recap on the general overview of vitamin. Recall that vitamins are generally classified into two. You have the water-soluble vitamins and the fat-soluble vitamins. Their concepts are based on the name that it is being given to, as water soluble and as fat soluble. It means that those ones that are water soluble only dissolves in water, while those ones that are fat soluble only dissolves in fat. Now, the fat soluble includes vitamin A, which is what we are treating today, vitamin D, vitamin E, and vitamin K. They are fat soluble. While water soluble includes vitamin B complex and vitamin C. Vitamin B complex begins from vitamin B1 to vitamin B12. Now let's dive into the class. What is vitamin A? What is vitamin A? Vitamin A, as we all know, is a fat-soluble vitamin. It has a bioactive form that is present in animal tissue known as the retinol. While another type of vitamin A that is found in plant tissue known as the beta carotene. So we have vitamin A. It's been divided into two. The retinol, which is found in animal tissue. We have the beta carotene. This beta carotene is found in plant tissue. So this is for plant tissue, while this is for animal tissue. This beta carotene has a double beta ionion ring. It has a double beta ionion ring on its structure. As you can see in this structure, this is the structure of beta carotene. It has two beta ionion ring. This is one and this is two. This ring is known as the beta ionion ring. While retinol, as you can see here, it has a single beta ionion ring. So this is beta carotene, beta carotene. Why this is retinol? This becomes retina. Why this becomes retinoic acid? Retinoic acid. We are going to come into these diagrams later. Okay, we have given a concept about this. Vitamin A, which is called the retinol, occurs in three forms. It occurs in three forms in the body. Number one, it occurs as a retinal, that is the vitamin A aldehyde. Retinal, vitamin A aldehyde. Number two, it occurs as a retinol, that is vitamin A alcohol. Vitamin A alcohol. Lastly, it occurs as a retinoic acid, that is vitamin A acid. Retinoic acid, that is vitamin A acid. This is the three forms in which vitamin A occurs: as a retinal, as a retinol, as a retinoic acid. Now, it has a basic interconversion because all these states of vitamin A are interconverted. Okay, the first state of vitamin A becomes retinal. Retinal is converted to retinol. Retinal is converted to retinol, and it is a reversible reaction. Retinol by an enzyme called retinal reductase enzyme. Retinal reductase, which requires NAD H plus H as a coenzyme. NAD plus NAD H plus H. Okay, since retinal has been converted to retinol, retinol will be converted to retinoic acid. That step is an irreversible step. It does not reverse as retinol will be converted to retinal. Recall that this is the aldehyde form of vitamin A. This is the alcohol form of vitamin A. It goes this way to form retinoic acid by an enzyme called retinal dehydrogenase. Retinal dehydrogenase. This is how it works. So this reaction is an irreversible step, which is for this, why this is reversible step. Okay, the next thing that we'll be looking onto is the absorption of vitamin A. Vitamin A absorption. We have done our introduction. The next thing is vitamin A absorption. Before we delve into the absorption of vitamin A, recall that all the compounds that possess vitamin A characteristics or esteristics is called retinoid. All the compounds that possess vitamin A esteristics or characteristics is known as retinoid. We call them retinoids. Retinoids are those compounds that possess vitamin A characteristics. Okay, let's continue with the absorption of vitamin A. Vitamin A is absorbed in the small intestine. Why? Because the enzyme needed to break down the compounds of vitamin A, such as the retinal to retinol and retinoic acid, are found in the intestinal mucosa. And that enzyme is called retinal reductase, which is the first primary enzyme that breaks down the aldehyde form of vitamin A, which is the retinal, to the alcohol form of vitamin A, which is the retinol. Okay, but then before that process will take place by breaking down retinal to retinol, the provitamin of vitamin A, called the beta carotene, is being cleaved in the small intestine by an enzyme called beta carotene dioxygenase. Because when the cleavage of this compound occurs, it will give rise to retinal. That is why you are seeing the retinal compound here, because the cleavage of beta carotene leads to the formation of retinal, which is being catalyzed by an enzyme called beta carotene dioxygenase. Call it beta carotene dioxygenase. This is the enzyme that cleave what? That cleave beta carotene to give us retinal. That enzyme is called beta carotene dioxygenase. Recall that vitamin A is a fat, so it requires bile salt for it to be absorbed. And when it is being absorbed, it is being absorbed with other fats. And that bile salt helps to do what? Emulsify it before other fat enzymes will act on it. So it requires a bile salt. Requires a bile salt. It requires a bile salt. And there is also a presence of oxygen. Oxygen is available. Okay, since this beta carotene has been cleaved to give us retinal, this is the aldehyde form of vitamin A. The aldehyde form of vitamin A, as we know, the aldehyde form of vitamin A aldehyde form of vitamin A. This is it. Okay, this is the structure of retina. That is the first formation after the cleavage of beta carotene, which is being catalyzed by an enzyme called beta carotene dioxygenase. Because that all these things happens in the small intestine. Okay, now this is the functional group of the aldehyde. We call it retinal aldehyde. It is the vitamin A form of what? Aldehyde. We call it vitamin A aldehyde, which is the retina. Once retina is being formed, what will happen next? Retina will be reduced to what? Retinol by an enzyme called retinal reductase, which is what we did here. This is just like the chemical structure of it, give rise to, which requires a coenzyme, NADH plus H. NAD plus will give us NADH plus H. And that enzyme is called retinal reductase. We call it retinal reductase. We have formed our retinol. This is the vitamin A alcohol form. So this alcohol form of vitamin A, the alcohol form of vitamin A. Why this is the acid form of vitamin A? Because it contains carboxylic group of an acid. So this is the functional group, the alcohol functional group. Why this is the aldehyde functional group? This is the carboxylic functional group. That is why it is known as the vitamin A acid form. So here is the acid form of vitamin A. Acid form of vitamin A. This is how it goes. After the reduction, we get our retinol. From our retinol, we also generate our retinoic acid. Recall that the formation of retinoic acid is an irreversible reaction. Why the formation of retinol from retina is a reversible reaction. That is how it works. But after the absorption of vitamin A, where is it going? It is entering the liver. But vitamin A does not enter the liver straight. It has to be incorporated with chylomicrons and enter the liver. Once it enters the liver, it is being stored as a retinol palmitate. It is being incorporated as chylomicrons and stored in the liver as a retinol palmitate. That is a storage form of vitamin A in the liver. Is stored as retinol palmitate. Retinol palmitate is the storage form of vitamin A in the liver. That is how it works. So the next in line is what we call transport of vitamin A from the liver to peripheral tissues. That is the outline that we'll be discussing now. After the absorption of vitamin A from the small intestine to the liver, recall that we said previously that vitamin A is stored in the liver as a retinol palmitate. After it has been stored, it has to be transported from that liver to the peripheral tissues or the extrahepatic tissues. That is being championed by the retinol binding protein. It is the activity of the retinol binding protein to transport stored vitamin A, which is the retinol palmitate, as a transretinol, because the stored vitamin A, which is the retinol palmitate, is transported as a transretinol by a retinol binding protein. Retinol binding protein is very, very essential in the transport of vitamin A from the liver to the peripheral tissues. So up to the next we'll be discussing is the retina. We'll be discussing retina. You have the retina. Retina is essential for vision. It's very, very important for vision. So we'll be discussing the biochemical basis of the retina. Okay, the retina contains two photoreceptor protein or photosensitive protein, which is the rods and the cones. Contains the rods contains the cones. The rod is responsible for dim light vision, while the cones are responsible for color vision and bright light vision. The rods contain a photomembrane protein known as the rhodopsin. Contains a photomembrane protein known as the rhodopsin. Rhodopsin is a photomembrane protein, while the cones contains a photosensitive protein known as the conopsin. Conopsin is a photosensitive protein that is found in cones, while rhodopsin is a photomembrane protein that is found in the rod. This rhodopsin contains two important proteins known as the 11-cis retina and the opsin. The 11-cis retina and the opsin. This opsin is very important, and as well 11-cis retina. This conopsin is as well vital in color vision and bright light vision. Deficiency of 11-cis retina leads to night blindness and increase in dark adaptation time. When 11-cis retina is deficient, it leads to night blindness and increase in dark adaptation time. Okay, decrease in production of cones leads to what we call color blindness. When there is decrease in the reduction of cones, it leads to what we call color blindness, because cones are responsible for bright light vision and color vision. Okay, we have various types of cones. We have cones that are sensitive to blue, cones that are sensitive to green, cones that are sensitive to red. Those are the various types of cones. So let's delve into the next outline, which we'll be looking onto. That one is the biochemical importance of vitamin A. Biochemical importance of vitamin A. The biochemical importance of vitamin A, number one, the acid form of vitamin A, which is the retinoic acid, act as a steroid hormone. Retinoic acid act as a steroid hormone. Number two, retinoic acid regulates gene expression and tissue differentiation. Retinoic acid regulates gene expression and differentiation of tissue. Number three, biochemical importance of vitamin A is that vitamin A plays a huge role in vision. We call it George wide visual pathway. It plays a huge role in production of sight. That is George wide visual cycle. Another biochemical importance of vitamin A is that vitamin A is associated with anti-cancer therapy, which is mainly attributed by carotenoid. Carotenoid is found in vegetables. A vegetable that contains carotenoid is said to be important in the decrease of incidence of cancer. So when you take vegetable that contains carotenoid, it's said to be used in decrease in the incidence of cancer. So we have number 4, carotenoid for anti-cancer therapy. Anti-cancer therapy. Okay, these are the biochemical importance of vitamin A. Vitamin A plays a very, very vital huge role. It plays a what? A very, very what? Vital huge role. Okay. Another one we'll be looking on to, let's add it, is vitamin A is associated in keeping or the maintenance of retinal epithelium and skin, or it helps in the maintenance of normal epithelium and skin. So maintenance of normal epithelium and skin. These are the biochemical importance of vitamin A. Okay, let's dive into the applied biochemistry associated with deficiency of vitamin A. What are those things that could be implicated when there is deficiency of what? Vitamin A. We have number 1, let's term it applied biochemistry. Applied biochemistry. We have number 1, which is noctilopia or night blindness. Noctilopia or night blindness, it is a decrease in visual acuity or diminishness in the visual acuity, which is as a result of increase in dark adaptation time. It could be associated with the deficiency of 11/6 retina, because when there is deficiency of 11/6 retina, it will lead to what we call night blindness or increase in dark adaptation period or time. So noctilopia can be called night blindness, which is what I've explained in respect to deficiency of 11/6 retina, which will lead to night blindness and what? Increase in dark adaptation time. The next thing we'll be looking on to here is xerophthalmia. Xerophthalmia is a condition whereby the conjunctiva of the eye becomes dry, thick, and wrinkled. When the conjunctiva of the eye become dry, thick, and wrinkled, it is safe to say that yes, the conjunctiva has become what? Keratinized, thereby losing the transparency of each state, because it is always transparent. So when it becomes what? Keratinized, it will lose the transparency state. Automatically, it is now dry, thick, and wrinkled. And the dryness could be escalated to cornea, thereby leading to corneal dryness. And once cornea become dry, the cornea loses its epithelium. So it is associated with what? Deficiency of vitamin A. The next one is bitter spot. Bitter spot. Bitter spot is a condition whereby there is increase in thickness of conjunctiva in a certain area of the eye. It appears as a grayish white triangular plaque that is firmly adherent to the conjunctiva. So it occurs on that way. I believe if you see a patient that has bitter spot, it occurs as a grayish white triangular plaque that is firmly adherent to the conjunctiva of the eye. So it occurs as a result of increase in thickness of the conjunctiva of the eye. That is the bitter spot. The next one is keratomalacia. Keratomalacia. This keratomalacia is associated with persistent xerophthalmia or a progressive what? Xerophthalmia. That is what is majorly caused with, because when this xerophthalmia progress, it will lead to what? Keratomalacia. All these things could be reversed with vitamin A supplementation. But do not take excess vitamin A to avoid hypervitaminosis of vitamin A. We call it hypervitaminosis A, which is what? Vitamin A toxicity. Because excessiveness of everything is not good. Do not use, because yes, if you give vitamin A supplementation, these diseases could be reversed. Do not take it too much. It has regimen, it has range in the body. So that these are the applied biochemistries that is associated with vitamin A deficiency in the eye. The next thing we'll be looking onto are those things that causes vitamin A deficiency. Causes of vitamin A deficiency. Causes of vitamin A deficiency. Vitamin A deficiency. What are those things that causes vitamin A deficiency? Number one is decrease in vitamin A intake. When you have a lesser quantity or a lesser consumption of vitamin A, it will lead to what? Vitamin A deficiency, because it will not be enough. The next one is obstructive jaundice. When there's obstructive jaundice, there will be defect or defective absorptions of vitamin A in the small intestine. That is why it is one of the factors that causes vitamin A deficiency. The next one is liver cirrhosis. When there's liver cirrhosis, there will be decrease in the synthesis of retinol binding protein, that is RBP. Very, very vital. The next thing that causes a vitamin A deficiency is malnutrition. When there's malnutrition, whereby there is no availability of an amino acid to produce retinol binding protein, automatically it is a problem, because that retinol binding protein is essential in transport of vitamin A from the liver to the peripheral tissues. The next one that we'll be looking onto is chronic nephrosis. When there's chronic nephrosis, there will be higher excretion of retinol binding protein to the urine, which is very, very fatal. So here comes the end of this class. We have discussed the biochemical importance about the retina, the structures of the beta carotene, the retina, the retinol, and the retinoic acid, which is the three states in which vitamin A occurs. I believe we have done justice to this class. If you have any question, write it down on the comment section. Endeavor to share this video to your colleagues or to your friends in medical school. Subscribe to my YouTube channel and anticipate for more videos like this. Thank you for watching.